So I want to seize this opportunity to welcome each and every one of us here again. For those of you in the earlier session, I believed you had fun, and to those who are joining new, you are welcome to the next session of the community, Power of Community Talks. And during this session, we're basically going to hear from some amazing speakers and some wonderful stuff they have been doing in their respective countries. So I wouldn't want to waste much time. I want to introduce our speakers, and they will take turns to introduce themselves when they are given the opportunity to present what they are doing. So as part of the Power of Community Talks today, we have Diana Sophia, pardon me if I don't get the pronunciation correctly, Diana Sophia Molokana, who is working at the Plant Biotech Lab and other representing other organizations. We have Nini Ortiz, who is affiliated to Robert O. Becker Lab in Madrid, Mexico. We have Mohammed Atif, who is at Peshawar Biohacking Space. And we finally have Rachel Aronoff from Aquarium. So we will take turns to listen to some amazing works that these speakers have been doing through their presentations. We will start off with Diana. Diana, over to you.
Thank you very much, Harry. Well, hi everyone. It's really great to be here. My name is Diana Mayocana. Actually, Harry, you did it quite close. Thank you. And I'm so excited, as I told you, to be here at the Bio Summit 4.0. I'm right now praying to be back together soon in the future, but at the same time, I'm very glad that we can be virtually together by means of technology. A little bit about myself. I'm a biotechnology engineer, born and raised and based in Quito, Ecuador. Greetings from South America. I work, as you can see, for the academia mainly as a plant biotech lab as teaching and research assistant at San Francisco de Quito University. And at the same time, I work for Catalysis magazine. This is the first bioscience digital magazine in Ecuador. And my approach to do-it-yourself bio has been by these two flanks, by developing teaching tools for students of different ages and by science communication. Today, I'm very happy to talk about the core. I will call them the fundamentals, the very reason of the appearance of the do-it-yourself bio movement, and those are equity and sustainability. And for doing that, I invite you to see it metaphorically as a DNA double-strand sequence. Next slide, please. It could be linear, supercoiled, it doesn't matter. You just have to use your imagination. Could you please, Harry, help me to share the screen with the next slide?
Harry, you're muted.
Yeah, apologies for that. I think there were some few challenges here, so I'm going to reshare my screen. All right.
The next one, please.
Okay.
Thank you so much.
You're welcome.
Here it is. Okay. There is the do-it-yourself biology DNA. Linear, super cool, it doesn't matter. You just have to imagine it, okay? But if you zoom in, you will see the code of it, the nitrogenated bases that integrate something that in the future will pass to transcription, and those are the concrete ideas that we may have. And after that to be translated to proteins, which in the metaphor are the products that we are going to produce. So Without that code of conduct, nothing will be based on the principles of equity and sustainability we're talking about. And those are open access, transparency, education, safety, taking care of the environment, gaining peace. In this case is actually gaining more than pursuing it, and tinkering. Those come from the 2011 North America Do-It-Yourself Biology Congress as a code of ethics. And what is notable is that as a double strand, they must have their complementary basis, which means that you have to design your ideas based on that, and your collaborators need to think likewise, with the same code of conduct. Now, if you sum up, you will see that our nitrogenated bases have three parts, just like in real biology. I call them the DIY biofoundationals, and they are open source culture. That is, don't be afraid that the others will steal your idea and will make more money than you. Don't be led by the money. The other part is the reenactment of a community of equals. And finally, it is to be conscious that we are in an unequal world, but we need to stand in the equity principles. Sadly, as you can see in the top right corner, there are some nucleases and enzymes over there that are willing to put these fundamentals to risk and threaten to fragment them if we enter into the metaphor. And those are inequality as the origin of unsustainability, which means that we do not care in wasting many resources or producing a lot of lab trash just to achieve our objectives. Other nucleases, unequal access to capital, knowledge, and technology, seeing that we're not doing a thing to change it as long as we are okay doing our sciences. The third one, I will see that is a lack of diversity in our teams, in all the aspects, not only gender, important gender, but there are other diversity types like demography, ethnics, even class. This is the most risky one, I will say, because sometimes we are not aware of them. We tend to surround by people that think like ourselves. So once we have considered this, what can be the path of our do-it-yourself bio projects? The first, and maybe the most obvious, get the lab outside the lab and carry it to those who need it. I got this experience by designing a portable lab to teach transient transformation, and I had the fortune to present it actually last year at the BioSummit 3.0. It has been a rewarding and satisfactory experience by using quite common lab materials, tobacco mold plants, bacterial solutions, and this also helped us during pandemics to do some essays for projects in the academic lab that I was working in. That was quite interesting. The second part is tell your science story, but not only in paper, but also in different ways. What about an online lab notebook, a blog, and so on? That's the reason why Catalysis magazine was born, with a group of 5 thrilling enthusiasts and great friends to communicate science in a friendly language. We started practicing to translate famous articles from Nature or other scientific magazines to most known words. That was specifically useful during the pandemics, where a lot of people thought that the virus was designed in the lab, to explain to them why that asseveration was nothing but based in fear. Finally, the approach of One Health, the third but not less important path. One Health means the health of the environment. It's the health of humankind. Because if the planet is sick, we all get sick too. I think I don't need to say more about this due to a current global context, but that gives us a message. The solution to a problem needs to be considered from all the environmental points of view. A clear counterexample is the use of squalene as an adjuvant for COVID vaccines. Squalene comes from the liver of sharks. Could it be justifiable to sacrifice millions of sharks to extract squalene and save our lives and buying it and making vaccines in our community labs and saying that it's good because it's not making by big companies? I don't think so. We can see that there is in fact difficult times, but I will not count them as uncertain because the power of community is what brings us close to the certain path, especially in the field of biology and biotechnology, which are growing quite fast. This fast march of things can make us sometimes lose perspective. Why are we doing? What are we doing? Maybe it's difficult to answer this, but I'll tell you something. That is, although we do in whatever field, but especially in the biodisciplines, has to be led by a personal and powerful purpose, which is to impact in a positive way the lives of others by considering what they really need and not only what we think they do. That's why it's important to promote what I consider the most important characteristics in the DIY biology field, is that the power of ones becomes the power of all. For that reason, the name of this talk is Do-It-Yourself Biology Tools for Equity and Sustainability. Before the technical tools, you need in fact to know the perspective tools. Thank you very much. I think we cannot hear you, Harry, but thank you.
Thank you very much. I think this was a great one. I think this is one of the very brilliant presentation we've had. I think we will take turns to listen to the next speaker, just after which we will then go into question and answer session. And I think let's be reminded that each speaker has 10 minutes of presentation, and we will then zoom into questions and answers towards the end of this presentation. So bear with us. If you have any questions, please note them. We can get interactive in the chat aside that while we wait for the Q&A session. So I'll take turns to have the next presenter, who is Nini Ortiz.
Hi.
Please do introduce yourself and the setting which your work is situated. Thank you.
Well, hello everyone. My name is Nini Ortiz. I am from Merida, Mexico. I'm starting up a lab, a DIY bio lab, but I am trying to create a new concept that I haven't seen much in the DIY bio community, which is called Bioelectricity. I am not sure if I can share a presentation I prepared.
Yeah, please do. You have you can share your presentation. So I'm just going to stop sharing.
Okay, I have 10 minutes, right? Okay.
Yes.
Thank you. Yeah, well, this is the presentation I prepared for you. It's titled How to Develop a Reproducible Bioelectricity DIY Biolab. So I can't move it forward. Ah, why I can't move the presentation forward? Ah, here it is. So what is bioelectricity? What I refer when I say bioelectricity? Bioelectricity to me refers to the ion multiforces that arise in cellular membranes thanks to differences in ion concentrations from charged particles. These differences in ion concentrations are also generated through specialized proteins that can make the membrane less permeable to certain ions, so you can get more positive or negative intracellular concentration of ions. These differences in permeability generate patterns through development and regeneration. And recent research has found that they enforce the regulation of genes during development, and you can actually change the polarity of the regeneration of an animal, like a flatworm, just blocking a special type of junctions that change the overall electrical pattern of this worm. Just changing them, you can get a different phenotype. For example, the worm has a head and a tail, you cut it, then it regenerates and generates supposedly a head and a tail, but if you block the gap junctions, you can get a different electrical pattern. And this pattern somehow changes how genes are regulated. And afterwards, the worm will have a double head phenotype without actually changing genes specifically. Without tinkering with the genes, you get a global anatomical outcome, which is really, really interesting and new. And I haven't seen anything quite close to this in the biohacking community. It's not the only application it can have. If we understood how these gene regulatory networks are regulated by bioelectrical patterns, we could maybe somehow in the future generate more specific interventions in molecular biology. So you could just create new types of animals or forms, or anatomical forms. There are other examples I could use, but I will take a lot of time to explain, so I will continue. So what do I refer when what should a DIY biolab related to bioelectricity be? It should be a lab focused on research and innovation and education too, in these three branches of knowledge that are developmental biology, regeneration, and cancer. We want to approach this by conducting computational analysis and modeling, and through experimental lab settings with doing prototypes and different stuff, because in this field, in developmental biology, there are some reagents or equipments that are quite expensive to have. So the cheapest way is to prototype it. And these are some example model organisms that can be used to explore regeneration and development. And what is the mission of the lab I'm building with my group of friends that are also all biotechnology related people? We believe that bioelectricity is a really exciting new field that should be open to everyone. And we want students or any person actually that shows an interest in this new field of biology we want these people to come to us and learn and just follow their imagination and see what they can discover, because it's really an emerging field. So there is few labs actually doing this research. And in the future, we would love to grow and be a fully development lab. And we want it to become like a maker space for people to go and explore how bioelectricity can relate to their chosen species or organism. And I forgot to mention, bioelectricity is similar to neuronal impulses, but it operates in different time scales. So any animal can actually show some sort of a bioelectrical pattern. We don't know. There is not enough research to know how it happens in many organisms, but if we knew, we could get very powerful insights about how morphology arises in multicellular systems. So how can we measure bioelectricity? The traditional way is to use a patch clamp machine, which is really expensive. And new research has made possible to use fluorescence microscopy, which can be which is also expensive, but it can be made cheaper if you prototype it. And these are we to view bioelectricity, we use these fluorescent dyes that respond to hyperpolarization or depolarization signals in tissues or even in plant tissues. They are really wonderful. You can predict bioelectrical patterns using this computational engine that is made by Tufts research academics, and it's called Betsy. It's really awesome. You can, if you have the information of the membrane permeabilities of a tissue, you can put those as inputs in the program, and you can measure how this pattern will change through time. And what do you need to start a DIY Bioelectricity Biolab? I will suggest that if you can, you could get a stereo microscope. You can use an INITC fluorescence adapter to change it into a fluorescence microscope. You will need a good computer for computational analysis, and you can invest in basic research lab equipment like micropipettes, autoclaves, and you don't need too much stuff to do it. The reagents that you need to measure fluorescence are about $416. It's not that much of a big investment, but so it can actually be done, but nobody is actually doing it. So we want to be the one of the first people doing this. What projects are we working on? We're working on trying to make the NITC adapter cheaper than it actually is. So everyone could buy a stereo microscope and could get to our GitHub page, see how to do the prototype, and make their own fluorescence lights and filters for their stereo microscopes. And we're working on a model for film regeneration in zebrafish. We want to model both the computational analysis and do the lab experiments to see if we can get a match between the predicted and experimental stuff. And we want to make it sustainable. So we're at a multidisciplinary team. We're working on making workshops, talks, and seminars that expand on how bioelectricity can be used, how can you learn it, and all of that. And this is who we basically are. We are all friends, and we are all focusing on different tasks to carry on this project to fruition. And we are just starting. COVID has made it a little bit more difficult to get to a target community, to invite them to do workshops, because here it's pretty bad. We have lots of cases of coronavirus. But if we continue, we plan to continue this, and in the future, we hope we will finally be able to reach more people and expand on this new area of developmental biology and regenerative medicine. Thank you. Thanks to all of you.
Thank you very much, Nini. I think this was a great presentation and a very huge insight into your work with bioelectricity. Like I said, we would zoom into Q&A soon after the presentation. So please bear with us. If you have any questions, we would get back to those shortly. So I would take time to invite the next speaker, who is Mohammed Atif from Biohacking Space Peshawar. If you are here, Mohammed, please do let me know if you want to share your screen, or I can go ahead to share mine. Firstly, if we have Mohammed, can we spotlight him?
I know he's here. I think he's...
Okay, Assalamu alaikum. Greetings from Pakistan. This is Mohammed Atif. Do you hear me?
Hi, Mohammed. We hear you. So excited
Yeah,
to
yeah.
have you.
Thank you. I'm also very pretty much excited, and there's a problem with my camera. It's actually a night time in Pakistan, and I'm sitting in my garden and delivering my lecture and my presentation to you guys. And I'm very grateful to be here with you guys. Okay, so let me start from my introduction. I'm actually an undergraduate student at University of Peshawar, and for now, I'm working in DIY and biohacking, and it's actually a revolutionary idea for the people of Pakistan and for people specifically for my community. It's a very new idea, and as my title indicate that we are actually working to bring the idea of biohacking and DIY to our community, and what were the hurdles we faced during pitching of the idea. So, the first problem, it was to change the mindset of the community. And in our community, the students are actually pretty much involved in the theoretical concepts. Actually, uh, they are actually pretty much majorly they are focusing on the theory, not on practicals. So first we have to change the mindset of the students that practicals are very much important. And for this purpose, we build a website in the name of Biohacking Space Peshawar. We start sharing blogs with the students that about the recent updates in biotechnology, the recent updates in microbiology, and in specifically in the DIY and biohacking. So we address the problem of the change of the mindset of the student by introducing the blog post, introducing Facebook blog posts and Twitters, etc. you know and the second problem we are facing, as I am from the department of University of Peshawar, the teachers are actually they are actually believing that it's impossible for you guys to bring the idea of biohacking and DIY to Pakistan, because you need well equipped equipments and you need a good mentor. But despite of all this, we are working on an imagination. We are, there's a bunch of videos are right out there in the YouTube. You can learn from that, and you can apply and from where you are. So we just to convince the teachers at our department, we just started working on an incubator that are not working in our department. And it was very amazing. It was very amazing for the teachers that we repair the incubator that is not working in our department. And after this, the teachers are pretty much happy that you guys are doing a great job, and they get did they give us the project, and granted about 1 lakh Pakistani rupees for working on the other incubators and microscope. And the most important thing that we are facing is in Pakistan is the community, specifically the biological community, the bio related community is non-recognizable. uh, the people right out there, no one is actually reaching out to Pakistani students, and specifically from the students just like me. I'm from the very remote area. uh, they are actually the students find pretty much difficulty to reach out the foreign people to collaborate with them. And the Bio Summit, actually this provide me the platform to reach out such enthusiastic biologists and to share my ideas with these people. And it just motivate me, to work more passionately and more enthusiastically about the DIY and biohacking. And to get project for from the departments and from the organization, we must have to present some practical works. So we do we just build up a incubator that actually we just insert a thermostat, a light bulb, and a fan, and we just build up an incubator, and we present them to the, they give us the money. We give him about 10 to 15 samples to the students, just to promote the DIY, just to promote the biohacking. uh, as I told and as I mentioned earlier, that the practical importance of no, the practicals are not that much important in our society, just because that they think that if you do practicals in your lab, they don't give you the money in the future. This is actually the mindset that is built up in the student mind in Pakistan. So you know we told you, we built up different kinds of the seminars, and apart from this, we started taking classes with the high school students, to tell him that there is a world beyond, just you are doing ritualization of the subjects like what is biology, and this, there is actually a world beyond that. And we started working on this, and it's going pretty well. And our future project is actually is to focus on the high school students, and to bring them into the world of DIY and into the world of biohacking. That's it from my side, and if you guys have any question, then let me know.
Great. Thank you very much, Mohammed. I think this was really brilliant. I think one of the things that you said, which resonated with the presentation from Diana, was that tell your stories, your science stories, not only in papers, but via blogs and magazines. And I think you are doing it amazingly using websites just to connect with other young people like yourself, just to democratize what you are doing. Thumbs up with question and answers. We would hold on to that towards the end of the session. And I think we have finally an amazing speaker, Rachel Aronov, who would be doing us the next presentation. Rachel, do let me know if you have to share your slides, and I can stop sharing for you to go ahead to share yours. Thank you.
Yes, I think it would be best if I can share my screen when I get on there, because...
Please go for it.
Okay, so thank you very much for the opportunity to speak here at my first Bio Summit gathering. I'll go ahead and try and share the screen, and I can select a window even. So does it just show up nicely on your screens?
Yeah.
So this is a Prezi. So if anybody gets car sick or seasick, just look to the horizon. There's nothing to worry about. But I've been really looking forward to joining in on this, because I heard about Bio Summit in around 2017. One of our Hackarium members went to Boston for it, Dan Hernandez. And he was saying what a great experience it was. And then I met David when he came for the big event at the CERN. And I've gotten more and more into the whole DIY bio thing, especially because I had made my own association called Agir, which some of you have seen this exclamation point. So Agir stands for Action for Genomic Integrity Through Research. And it's the idea of genomic integrity is to take all the complex and dynamic genetic details of cells and put it into one short phrase. So people don't get too carried away with, oh, it's too complex, I can't think about that. And so with this association Agir, which I started around 7 years ago, I was trying to think about ways to promote research into the idea that there are risks to our health out there, and some of them we actually can choose to avoid. And so I was trying to figure out good ways to really demonstrate that some of the common things we choose to do, like things like sun tanning or smoking or dyeing your hair, are all things that might be better avoided, not just for us, but for the environment, for everything alive. And so when I heard about biohacking, I already mentioned this earlier today, it had to do with my work and a biosensor course that I put together. And actually, Carolyn Angleton was one of the participants in that course, which was held just at the end of January 2016. And I said, well, Hacquarium, these biohackers, I've done some soldering. What else can I do? Maybe I can run my experiments for Agir at Hacquarium. And so that's how I started in this whole biohacking world. And I started working towards participatory research for change. For a long time, people have talked about citizen science, but there are some difficulties about that because sometimes it's more like, well, taking advantage of people for free labor. And so we really like the idea of participatory research. And of course, because of Agir, my focus on public health and avoiding risks to health. And because of this session, sustainable biohacking is the long-term aim, that we can have these sort of community labs that are going to last in the long term and will be something that people think of as a good resource for education and fun too. So I'll just jump right into this very old slide that when I used to only do PowerPoints, we used this one. And the main point in Hacquarium, in the French pronunciation, it's aquarium. Oh, I think I already mentioned this also. Transparency. It's the whole DIY bio ethical code that we follow. But really, the people make the magic. La communauté fait la magie. So you can see here some of the very founding members, Luke Henry, Jan Pearson, Robin Schiebler. They were all there when I came to the lab the first time in, I guess it was November 2015. And I was so impressed in doing the soldering, and here's Sam from a group called Octanis that was started actually as a Hacquarium project and became, now it has its own association, Octanis, and a startup with its own branch office, not only here in our co-op in Ecublon, Switzerland, but also in Zurich. They have a second base. But learning to use, we were doing the laser cutter there, me and Sam. And this is Vanessa, one of our most famous bio artists. But basically, over the time, these are some more recent pictures of a few people. There's Tatiana and Raphael, Tatiana and Vanya. And we were using this heat sensor to try and see how his breath passed through, like for seeing how good masks work. We were doing a little mask experiment. And this is our last, just from last Wednesday, actually, we had a big assembly general meeting and there were 4 participants on Zoom, a few people that also gave their little signatures that told us what they wanted to vote for. And Hacquarium, in terms of becoming really sustainable, we're trying to get recognized as an association of public utility. So like tax deductible status. And but of course, Hacquarium has always been around different projects. And so this shows our biomusic group, Living Instruments, that I described to somebody else today, our yeast organ, which is shown here only with 5 cultures. They're all fermenting. And as the bubbles of gas go past these sensors, it's hooked up to a synthesizer and you get out different sounds. For this concert, they actually had a real instrument, a flute, but it was going through some interesting bubbly thing also. And this over here is Vanessa with her musophone. So the moss has capacitance sensors inside of it so you can stroke it and get music out of it. So these guys have given world tours in London, in Paris, and then some of our workshops that we've done over the years include this Open Food Repo DNA workshop where we did Minion sequencing of food ingredients to compare the foods, what was on the ingredient list to what came out with the sequencing with the Minion. And that was in collaboration with the EPFL, one of the universities near us, and Swiss Decode, which was sort of a spin-off of a Hacquarium project called Beer Decoded, which was about your beer. And actually, I did some work with Swiss Decode also, which is part of the reason I could contribute so much for our Corona Detective project that I'll talk about later. But I could talk a really lot about genomic integrity and Agir and all the important ways that DNA is not just set in stone like the sequencers want to tell you, but is actually very dynamic, and you need recombination and mistakes during recombination, for instance, to have your immune system work. But I'm going to try and quickly go through sort of the experiments we're doing for Agir at Hacquarium, which include open sourcing these really classic methods of DNA damage detection, like doing the COMET assay. So we're using inner cheek cells, and this shows some cheek cell comets. The nucleoid, all the leftover DNA and RNA that was within the cells shows up as the sort of the head of the comet, and the brighter and stronger this long tail of the comet, the more double-stranded breaks you have in the cells. But maybe for some of you, you'd like to go back a bit. What is this, a comet? Are we going out to the stars, or are we going down to the cells? Well, I should just say quickly, cells, of course, are our body's basic building block. And, you know, it's full of DNA, but RNAs are important. There's epigenetics, a whole bunch of translational modifications of different things, little small RNAs changing gene expression. And just organizing a cell division, as this cell is about to divide, it's already duplicated its DNA, it's going to do mitosis. Some of you probably already know that the DNA in our cells is 2 meters long. So to organize that for proper cell division is quite tricky. And if you zoom in even further into the cell, then you see you can get the idea of the nucleosomes and how complicated it must be for gene expression. So here's the DNA and 3 nucleosomes along here. This is work of Drew Berry that he submitted for the Agir Art Call, which is still open if there are any bio artists that want to show us what you think of as the molecular dance in the cell. But basically, the reactions happening in cells are all mediated by enzymes. Some of them are RNA enzymes, and most of them are dependent on common salts like magnesium or trace elements like iron. So just to zoom in on an active site of one enzyme with its coordinated magnesium, actually, people don't always realize, but ATP itself wouldn't work if it wasn't coupled to magnesium. So I think that's one of the things a lot of us are missing in our diets, and I don't usually recommend any supplements or anything, but enough magnesium is important, and they know that for a long time. So I'm not going to go through all the details because I'll go past my 10 minutes, but basically we know that breaks in DNA are something that you should really avoid to protect your cells from cancer development. And so even though I think of DNA damage as just a very small fraction of what genomic integrity is, to look for these in an open source way to allow people to look for it in their own cells seems like a really good idea. And I've been really successful with micronucleus assays, and we're working towards a cheek cell chip. So the typical micronuclei assay means we just make some slides, and we use a toothbrush to collect our cheek cells in saline solution usually, and we smear the cells out onto microscope slides. We stain them with methylene blue, and most cells have one nucleus as expected, but every now and again you will find a micronucleus. And a high frequency of micronuclei is really correlated with increased risk of disease, especially cancer. But the first thing I always have to say in these workshops is, if you see a micronucleus, don't panic. And the idea is that people get an idea of how the scientific method really works and how statistics are important. That if you have 5 or less micronuclei in 1,000 cells, is totally normal. DNA breaks, it gets repaired. We have over 100 genes in our chromosomes that repair DNA. But if you have 20 to 50 micronuclei in your 1,000 cells that you count, then maybe you should think about, is your DNA repair faulty? Or perhaps are you exposed to something? You've been taking too many flights? Have you just had an X-ray? What's going on? Maybe you should see your doctor. So it's all about participatory research, and these workshops are super fun. I have a little time-lapse video actually of this workshop, which was from May 2019. Oh, it seems so long ago. I mean, for my workshops with the toothbrushes and everything, I'm not quite sure how we can modify this. I have one thing to say about that, but at the end we can watch this time-lapse if you like. And of course, with the virus, we couldn't do quite the same things, but my idea is that perhaps people could go home with a couple of microscope slides and droppers and saline solution, and we could teach them how to just make the slides and dry them down, flame them, and then bring them all clean for the workshop for staining the cells and scoring. But we're still obviously waiting. So now I'll tell you, the long-term dream really is that we develop a cheek cell chip. And the idea is that instead of doing like our COMET assay, normally I use an old laboratory electrophoresis chamber, and I just have my cells embedded in agarose. Well, in this case, we're hoping to prototype a real chip that would do both the micronuclei and then the COMET assays on the same population of cells using the open flexure microscope. And I learned about these when I went to Shenzhen, to Gosh, and we did a nice epifluorescence build for the COMET assay. And I must say, so far, I haven't yet seen comet tails with the epifluorescence on this little 3D printed microscope, but we have seen nuclei already. So I'm pretty confident that when we get the better Raspberry Pi camera that's a bit more sensitive, we'll be able to see the comet tails too. And the idea is that we'll, on the same, there are motors here. So it's a motorized stage, it's high precision. So you can take a panorama of the complete sample, first for the micronuclei and then for the comets. And then finally, you'll be able to understand about the frequencies. And so this is my little area to talk about panoramas, because you can imagine this is just a view from my neighborhood. There's the Alps in the distance. Mont Blanc is hidden behind this hill over here. There's a moon right there. But you can imagine that a lot of squares of this panorama wouldn't be very interesting. But if you got all the data for your whole cheek cell prep in your cheek cell chip, you would be able to put it all together and perhaps even get automatic recognition to work. So this is one thing that Tatiana from the group has been helping me with. We've been trying to train a neural network to automatically detect the micronuclei using artificial intelligence. So here you can see there's one micronucleus here, and so these are the masks for the training. But you can also see here, there's this other spot. Is that a micronucleus or not? We're definitely learning by doing here, but the dream is that we'll be able to do easy experiments for biohackers everywhere with this automatic detection for micronuclei and comets, so that people will be able to assess their baseline levels of damage and then do some sort of experiment, either modify their habits or like say that you go running every day, you eat blueberries every day, and you see if the baseline level of damage changes, or you can do experiments outside of your body, ex vivo. And actually the first comets I showed you were treated, they're the positive control. They were treated with hydrogen peroxide, which makes tons of breaks. So normally you just get a little cloud behind the nucleoid. And the hope is that everybody together, doing it together, will participate more and act. And again, this is just to mention that learning by doing is a big part of all this. I've had a lot of interesting experiences in some of these workshops, people saying, I'm scared to see my cells. I already had a bone marrow transplant. Some people coming to visit at the lab here just maybe over a year ago. 3 people, we just did their cheek cells and we left them, we coded them. So it was blind. I didn't know who it was, but one person had this whole line of nuclei in a row. And so it's really been very exciting. And just before my time's all done, I just thought I'd mention a couple of other participatory research projects at Haquarium, mainly against pollution and for public health. We have the Montreux Clean Beach Project that is about water monitoring around the Bay of Montreux. Whenever that big jazz festival happens every summer, there's a big peak of pollution that comes into the lake. And we did 2 summers running, 8 weeks of sampling, and it was really great because this summer we got our no jazz control. And so we're putting all that data together, and it may well be we're going to go for a sort of academic paper because when we've been sharing all the data openly during the experimentation, we never really got that much feedback or when we offered to share a draft of the report to the sewage treatment plant people, we never really got a big reaction. So we think maybe we do have to try and publish a real paper. And there are other things about mercury, about lead, about other plastic pollution, and the corona detective that you'll hear about on Sunday. So maybe some of this is interesting to you and you'd like to join in for such transdisciplinary projects. And the aim is we get people with all kinds of experiences working together on the same projects. And you should all know that the Haquarium projects are for anyone to replicate, to hack, to modify, to share. And yes, if you tell us about what you're doing, we'd love to know, but you know, we can't force anyone, but we like the idea of doing it together. It's more fun than doing it by yourself. And I don't know, I didn't check the time. Did I actually go pretty good?
Yes.
We can show, should I go on to the time lapse? Here, there's the.
Sure. I think this is a great one. If you have some. So there's an echo. Can you hear me?
Should I get off my screen, actually? Sure.
If you might want to wrap up, we are actually moving into the Q&A session. So if the last slide, then that is great. I think this is an amazing presentation. Rachel and the other presenters, thank you very much for such.
Did you actually see my screen? Did you actually see my screen during all that? Yes.
Yes.
Good, okay. Because they just asked me a question, the Zoom, and I just got worried. Okay, fine.
Good. Very fantastic work that you are doing, and to the other presenters, very amazing work. I think this is where we pause and have it more interactive, where we ask questions. I know there are some very amazing comments and questions in the chat, but I want to open it up now for us to ask questions to our panelists, to our presenters, to hear from them what they have been up to. Any questions? So it is now open up. Thank you.
That was great, Rachel. Thank you. I'm sorry, I missed where Haquarium is physically located. Where are you?
Yeah, so we're near Lausanne, Switzerland. We used to be in a more industrial area closer to Lausanne called Renon. And now we're in a little bit more suburban area closer to the EPFL and the University of Lausanne.
Oh, how lovely.
We're actually in a very non-sustainable space right now because we took a building that's destined to be destroyed in the next 3 to 5 years. But that means it's quite low rent and it's just all one floor, sort of has a clubhouse feel. But yeah, we're on the lookout for a real space where we can move to before they tear it down over our heads.
Well, I hope your old building runs the same way our old building did. We moved in 2013 into an old building, and they gave us a great rent deal because same thing, they didn't know if it was going to be around, and now they've decided to keep it around and kept us.
Oh, wow. So yeah, it could well be. We'll be here for a long time. Yeah. With the corona, of course, we couldn't, if we had a much bigger space, it would be just a waste.
Amazing. Do you have? I think we have one question for you, Richard, in the chat, which says your video on the biomusic was awesome. Awesome. Do you have it on YouTube? I think that was a question.
Yeah, definitely. If you Google living instruments, you'll find something on YouTube. And also in the Hackarium Wiki, I think there are some links to things. There was a real composer. I mean, one of the first, was it? Yeah, when we did this biosensor course, we, no, it wasn't with the, anyway, they've done concerts in numerous locations. And the last one in Switzerland maybe was in Bern.
Great. Please, do we have any other questions for our speakers? Any comments, any inspiring moments you want to share? Anything that you heard that actually struck your mind and perhaps resonated with you? We would want to hear that.
There was a question about
Hey, what's going on? So I'm Tal. I was scheduled to be one of the speakers here, and then the talk was converted into a workshop. So just the communication on that wasn't entirely clear. So letting you guys know that the DIY tools talk that I was going to be giving will be tonight at 6 p.m. Eastern to 6:30, and that will be in a Nucleus, I believe.
Cool.
Brilliant. Thank you for sharing that, Tao. We will try to represent. Good.
I'll put the Prezi link too. Oh, maybe I won't. It doesn't want to.
Oh, thank you very much, Scott. I think there are very amazing comments that are coming. Very inspiring session, so much diversity in what people are doing. Thanks. Maybe some few more questions. I think we are getting close to the hour to have some break. So I think we might want to end it on a good note. So if you have any questions, some things you want to share that generally resonates with you, I think this is the time.
I wonder, is Scott, are you here? Did you say that you've already done this thing becoming a public utility?
Public utility?
No. Like a nonprofit.
So we're a provincial nonprofit here in Vancouver. So our province, they have their own rules. They're also federal nonprofits, but that's organizations that operate across provincial boundaries. So we incorporated back in 2015 as a nonprofit. So we're 5 years old this last June officially. In Canada, there are two types of nonprofits. They're just regular nonprofits, and then there are charitable nonprofits. So we don't yet have charitable status, so we can't issue donation receipts. But if anybody's working towards, and each country and area is different, if anybody's working towards that, if you can issue donation receipts, then there are a lot of people at the end of the fiscal year that want to dump money to get those receipts.
And support something they like the idea better than the government.
And so we're trying to work on that. The problem is it's complex in Canada, but Revenue Canada has made it a little bit simpler by putting a lot of the stuff online. Yeah. So each country is different.
So the meeting last week was about, we changed a few very slight things in our statutes to make it look better for our application, which I hope we'll get in by the end of November.
Oh, okay. Yeah. Here they actually, there's the BC Societies Act. So it's the government legislation on how a nonprofit society is supposed to operate. And they give a template for, or at least they used to give a template for what the bylaws are supposed to be. So we're supposed to have our own bylaws. It's governed by the statutory laws in the province. But what you have to do is don't just accept any standard template. Look to see what other people are doing. So for example, there's a nonprofit here that's a makerspace, Vancouver Hackspace, and they made the decision to get their have their membership decide membership fees, but the cost of everything in Vancouver has just been ramping up. Nobody wants to increase their fees, but they're desperate, and it has a real impact on. So look at details like that, but that's got nothing to do with biology. So.
Yeah. But it's about being sustainable too.
Yes. Yeah.
Yeah, I think the earlier conversations about starting and sustaining community labs and generally the set of presentations we have today are actually bringing to bear some of the challenges, so to say, and how our various unique settlements and where we find ourselves, we are using that to overcome the challenges that we face. And I think that is great. And that brings me to the feedback from Mark, and he says, thank you, presenters. The talks were impressive. You made me realize that any actions toward community bio counts, and that small ideas can be powerful when supported by community. And I think this is really great. And personally, I want to say that the work Mohammed is doing is very amazing. And we can only say that the community in which you find yourself is a great one. That we can support you in our own small ways. So keep doing what you are doing, and trust me, you would impact your community in a very amazing way that you'll be so amazed when you come back to see how you all started. Yes, there is, okay, I think Scott is asking a question. Derek, Scott, is that a question? Do you mind?
Oh, that was actually just for Derek. Maybe I should have done that.
That's fine. Our makerspace in Victoria is registered exactly the same as yours, as a nonprofit, not a charity.
Yeah. Okay.
Yeah. So I think I would also want to ask a question. We still have some time to our break. So my question has to do with the registration models for our spaces, because most of the time I hear you say that you register it as a nonprofit. Why are you not looking into a more profitable venture? Why are you looking towards a nonprofit or a charity that is not exclusively in for making money?
It's a good question.
May I describe our Victoria model and why we're remaining a nonprofit?
Yes, I think that's the question.
Yes, I was not on the panel, so I wanted to make sure. So we have some sources of income that are tied to our being a nonprofit, but specifically the relationship that we have with our landlord, which is an outgrowth of the University of Victoria, is that they have set us up in the back of their technology park, and in lieu of a fixed rent, we pay a portion of our membership proceeds, and they advertise our makerspace as being an amenity of the tech park. So they have things like a cafeteria and a gym to work out, and they placed our makerspace kind of on that same level so that when employers want to move into the technology park, they see the list of amenities the park offers, and our makerspace is right up in there with it. And so it's important for our arrangement with them, since the school is a nonprofit, that we also be a community-focused nonprofit. That is of much more importance to us than trying to make money on something that's really very difficult to make money out of, community spaces.
Good. Any more comments to that? Any additions? Why we are more focused to looking at non-profits rather than a financial, a money-making entity that is.
Yeah. So, from my perspective, it's a completely different dynamic. So if you are a corporate, a company entity, a for-profit entity, then your goals are very different from a nonprofit. And it's, for this type of stuff, it is often equally difficult to actually build up a corporate. But in terms of engaging the public, I think a nonprofit is an easier way to go. There's people that have mixed motives in terms of why they're a corporation. It's just very different, and there's the issue of trust. If you're a nonprofit, then there's a greater acceptance, at least by community, about what your motives are. Whether that's true or not, I'm not too certain. But I think there are certainly some labs that have set up as, instead of a public space, they're what's the term? Research, lab space for rent, essentially. They do well with what they want, but the public doesn't really access them. It's really for startup people wanting to have low-cost access to common equipment. And that's an okay model if that's what you want to do. But in terms of engaging the public, trying to expand the public's understanding of science, it's a bit more difficult unless you're one of the wealthy ones, where then you can fund community engagement. That's my 2 bits.
Thank you, Scott, for that.
If I could actually throw one more bit of elaboration in there on my answer. Makerspace itself is a nonprofit, but we actively encourage for-profit ventures to come out of the makerspace. And we've had 12 different individuals start small businesses out of the space. And one of the delights of a shared nonprofit space is that they can do so without owing any intellectual property or any fees or anything else to the space. So the space itself is a nonprofit, but individuals trying to make money and a living out of it is a very attractive model.
Yeah, and I love your model of doing a makerspace as opposed to just a biospace, because there's more community interest in a makerspace, whereas bio is very niche. And I think you've found that. We're a biospace, a separate nonprofit entity within a for-profit makerspace. And so we're just a renter. We have actually a really good relationship with Maker Labs, but still there is that difference in dynamics. So if people are there thinking about setting up a biospace, do think about making a makerspace and having the bio as a component of it.
Yeah, the original Hackarium definitely had the makerspace and Hackarium just side by side. Yeah. But after the big lab renovations, the makerspace was put downstairs and more startups kept coming in. And that's why they wanted our renovated lab and asked us for such a high rent, because they had people who wanted it. You know, there was just such a buzz. It was such a cool place to work. But once we were gone, you know, it's just not the same. I think. Well,
and the thing is, you lose access to the resources that a makerspace has that you can't afford within your own biospace. And
but so that's why we came together. We made a cooperative in the end with two other associations. So we have the makers that are welding everything, and we have the Octanis group that's all the electronics whiz kid type stuff and the laser cutter and 3D printers. So that's how I did the open flexure.
Yeah.
That's a great setup, Rachel.
Great. I think we are about to the last hour to this session. And I want to personally say it's been a great one and a very exciting one. I want to thank all the presenters for such an amazing presentation. Shall we give them a snap? A clap? Thank you very much. And we would go in for a break now and reconvene, especially for those of us who are interested in the breakout session, which will be coming on soon, featuring some amazing presentations coming in from other presenters on connecting local businesses together with fungi, how to create a closed loop ecological system. So if you're interested in that session, please do reconvene in about some few minutes, 15 minutes, 10 minutes from now or so. Thank you very much, and do grab some coffee and enjoy.